In the next section, we'll discuss major histocompatibility complex. So the major histocompatibility complex is also called the human leukocyte antigen, or HLA system. It's a closely linked group of genes on chromosome 6 in humans that code for a variety of immune-related proteins. Class 1 and Class 2 MHC proteins, as we have discussed, are involved in antigen presentation. It also encodes for certain cytokines and complement proteins, as well as other miscellaneous proteins that won't be discussed as part of our immunoseries. They're extremely polymorphic. Both sets of alleles are codominantly expressed. Two important points to remember about MHC. There are lots of variations of these MHCs, and that's what differentiates humans. That's what makes my proteins different than your proteins, my cells different than your cells. These are not hypervariable like T cells and B cells. When we talk about those being able to rearrange, that is not what happens with major histocompatibility complex. There are just multiple different alleles for each region, and therefore highly polymorphic. Both sets of alleles are codominantly expressed, which we'll get into a little bit later. So the alleles for Class 1 MHC are HLA-A, HLA-B, and HLA-C. MHC Class 1, as you recall, is expressed on all nucleated cells. It is involved in the capturing and presentation of cytosolic or endogenous peptides. The MHC molecule can fit fragments of 8 to 11 amino acids long into its peptide binding group. So the segments that are broken down from within the cell that are actually placed into Class 1 MHC are peptide chains of 8 to 11 amino acids. CD8 T cells are MHC Class 1 restricted because they are only activated when they recognize both antigen and MHC Class 1 in association. So when CD8 T cells are out looking for the cells that they need to kill, they're going to look at both the antigen for the specificity and the MHC Class 1 to make sure that it's being presented by the right kind of cell. So MHC Class 1 serves that function. MHC Class 2, the alleles are HLA-DP, DQ, and DR. Remember that these types of MHC are only expressed on what we call the professional antigen-presenting cells, the dendritic cells, the macrophages, and the B cells. These MHC molecules are involved in the capture and presentation of peptides that are processed from ingested vesicles, or in other words, exogenous antigen presentation. They can fit fragments from 10 to 30 amino acids, so a little bit longer of a fragment can fit into the peptide binding group on Class 2 MHC. Class 2 MHC is involved in presenting to CD4 T cells. So question, a deficiency in presentation through the major histocompatibility complex will not cause a change in recognition and subsequent function in which of the following cell types? A B cells, B T helper cells, C T cytotoxic cells, or D natural killer cells? The answer here is A, B cells. So where do we get the B cells from? Why is this the correct answer? Let's work our way through this. Remember that it's talking about MHC and will not cause a change in recognition and subsequent function. So which of these cells do not require MHC for recognition and subsequent function? We know that the T cells do, right? MHC has to present antigen to them, so we can get rid of B and C. So between B cells and natural killer cells, the NK cells actually look for MHC. Remember, they are looking for the expression of MHC on the surface of cells. B cells are trying to recognize antigen and its native conformation, antigen just floating around in the environment. Therefore, natural killer cells would also be affected by the lack of or deficiency in presentation through MHC, and the B cells then would be the correct answer. So here we have kind of a schematic of how the major histocompatibility complex breaks down. We see this complex, remember on chromosome 6, we said there's a group of genes. We call this the HLA complex. It's broken down into Class 1, Class 2, and Class 3. Let's speak about Class 3 first. So Class 3 has different regions. You can see C4, C2, and BF. And the gene products for these regions are certain complement proteins, TNF-alpha and TNF-beta. So we see some cytokines, some complement proteins being produced, but not typically what we think of when we think of MHC in terms of immune function. That's more Class 1 and Class 2. So let's dig into those a little bit. HLA Class 1 is broken down into 3 regions, B, C, and A. The gene products for these regions are a single polypeptide chain, HLA-B, HLA-C, or HLA-A. And this is where the polymorphisms come into play. So let's take region B, for example, and the gene product is HLA-B. Well, there are certainly a a wide variety of different HLA-B alleles that are available. And the ones that you have on the surface of your cells depend on your parents. You get one from your mom and one from your dad. So let's say you inherit HLA-B27 from your mom and HLA-B16 from your dad. That's going to be your those are going to be your HLA-B genes that you have, your alleles that you have, and that's what you will express on the surface of the cell, B27 and B16. Those might be yours. I might have B4 and B2 from my mom and dad. So you can see how you and I would be different as far as expression of the surface. It's not that we all have the same HLA-B and then it gets mutated into something different. It all depends on your inheritance. So the same could be said for HLA-C, and the same could be said for HLA-A. So as far as class 1 goes, every person is going to have on the surface of their nucleated cells 6 different HLA class 1 antigens, 2 B's, 2 C's, and 2 A's. For HLA class 2, same type of setup. You have DP, DQ, and DR. Now each of these actually encodes 2 different polypeptide chains, alpha and beta. And that's what makes up the class 2 MHC. And it's the same story. You're going to get a DP from your mom and a DP from your dad. Same with DQ, same with DR. So all of your antigen presenting cells, remember these HLA class 2 is only presented by antigen presenting cells, you will have up to 6 different possibilities, 2 DPs, 2 DQs, 2 DRs, each of those encoding 2 polypeptide chains, an alpha and a beta. And so let's look at what happens as far as class 1 and class 2 go. Let's start on the left. This is class 1. Remember we said that class 1 is involved in the presentation of endogenous antigens. So this starts with a protein antigen down in the bottom left that's existing in the cytoplasm. This protein antigen is broken down through cytoplasmic molecules that are involved in protein breakdown into smaller peptides that are then shuttled across the membrane of the rough endoplasmic reticulum and into the lumen through something called a TAP peptide transporter. Once inside the lumen, those peptides are loaded onto an MHC class 1 molecule. Now, once that MHC class 1 molecule has a peptide loaded in it, that's its signal to move out of the rough ER and into the Golgi complex. So we can see that happening as we move through a vesicle out of the rough ER and into the Golgi. Once it's inside the Golgi, then it gets addressed. Remember, the function of the Golgi complex is to tell proteins where to go. So in this case, it's going to tell this MHC class 1, you need to go to the cell surface. It'll package it, send it off in a vesicle, which will then reach the cell surface. Once it's on the cell surface, then it will sit there and wait for a CD8 positive T cell to come and take a look at it to see, again, if it's got the specific antigen and if it's the right MHC molecule. If both of those are correct, then that's a signal to the CD8 T cell that it should destroy this cell. Now, when do you want that occurring? Well, if there's a virus, for example, where do viruses make their proteins? Inside the cytoplasm. So that could be a viral peptide that's being loaded onto the MHC class 1, and therefore the CD8s can kill virally infected cells. What other types of cells might you want destroyed? How about tumor cells? This protein, this cytosolic protein, could be some type of tumor protein that would be recognized as by your immune system as foreign. Those can be loaded into MHC class 1, and they can be shuttled up through, presented to CD8s, and then the CD8 can kill off that tumor protein. That's how the CD8s can be involved in tumor surveillance. So there we have CD8s being involved in antiviral and anti-tumor responses. Endogenous pathway through the MHC class 1 presenting to CD8 positive T cells. Now, let's say that this cell is not infected, it doesn't have a virus, or it's not a tumor cell. Does it want to express MHC class 1 on its surface? Well, let's suppose that it doesn't. What may happen if MHC class 1 is not there? Remember that the NK cells are looking for decreased expression of MHC class 1. And so if the NK cell can't find MHC class 1, well, it may be apt to destroy that cell. If the cell doesn't have anything wrong with it, well, that's counterproductive. You wouldn't want that happening. So what your cells will do, if there's no foreign protein or no tumor protein to load into the MHC class 1 complex, it will actually load a self-peptide. We have plenty of self-peptides in our cytosol, right, that are always being broken down. It can load one of those self-peptides onto MHC class 1 just to get it to leave the rough endoplasmic reticulum and go out and be expressed on the surface so the NK cells won't kill it. Now, might a CD8 pick it up if it's expressing this? Hopefully not. Remember that most of the CD8s that would respond to self-peptides should have been eliminated in the thymus. So we really don't worry so much about expression of a self-peptide in the MHC class 1 because there shouldn't be a CD8 that will respond to it. However, the expression of MHC class 1 is necessary to prevent NK cells from responding. So that's all endogenous pathway, really cool type of pathway that our body uses. Again, antiviral response, anti-tumor response for CD8s, presenting self-peptides on that MHC class 1 if there's nothing else wrong in order to prevent the NK cells from becoming activated. Now, if we look at class 2 on the other side of the slide, now we're talking about an exogenous pathway. So remember, class 2 is expressed on APCs. These are all cells, B cells, macrophages, dendritic cells, all cells that like to endocytose things, bring things in from the outside. So it would make sense that this would be an exogenous pathway that presents through MHC class 2, right? So these cells will bring in exogenous antigen, be it through endocytosis or pinocytosis, into these compartments. And they will break down these antigens, these large antigens, into smaller peptides. Now we have to get it onto an MHC class 2. Well, the MHC class 2 comes from the rough ER, just like the MHC class 1 does. You'll notice in the slide that when it's in the rough ER, it has something called the invariant chain bound to it. Like MHC class 1, MHC class 2 needs to have something in its binding cleft in order for it to leave the rough endoplasmic reticulum. In this case, it's the invariant chain. Once that invariant chain is bound, it can leave the rough ER, go through the Golgi complex where it will get addressed not to go to the cell surface in this case, but to go to that endocytic compartment. It will leave the Golgi complex in a vesicle, and as it goes toward that endocytic compartment, the pH changes that are involved in the vesicle will actually digest that invariant chain, so that when it reaches the endocytic compartment, it has an open binding cleft that can then bind to one of those peptides that have been broken down from the exogenous antigen. Once it has one of those peptides in its binding cleft, then it will then be shuttled to the surface of the cell where the MHC class 2 can present to a CD4 T helper cell. So again, class 2, exogenous antigens coming from the outside being broken down, MHC class 2 coming from the rough ER with the invariant chain in its binding cleft. Once it leaves the Golgi apparatus, that invariant chain is digested within that vesicle so that when it fuses to the endosome, that MHC class 2 has an open binding cleft. It will bind to peptides within that endosome and then be shuttled to the surface where it can then express this antigen and hopefully be recognized by a CD4 T cell. So here we see the structure of MHC class 1 and class 2. Remember that we said class 1 is a single polypeptide chain, and in its conformation, when it's properly formed, forms 3 loops, the alpha 1, the alpha 2, and the alpha 3. The peptide binding cleft for class 1 is right in between alpha 1 and alpha 2, as shown by the arrows. It's important to note that there's another protein involved here with the MHC class 1 structure, and it's beta-2 microglobulin. This is not part of the MHC class 1 complex. Beta-2 microglobulin comes from a different region in your genome. Beta-2 microglobulin is also not part of what makes your self cells unique. It's not part of that polymorphic region. Everybody has the exact same beta-2 microglobulin. What it does do is provide scaffolding for the structure of class 1. And if you look, you'll be able to see that if that beta-2 microglobulin weren't there, then the alpha 1 loop wouldn't be forming. And if that alpha 1 loop didn't form, then you wouldn't have the peptide binding cleft. So although it is not specifically part of the MHC class 1, it's necessary for its structure in order to form that peptide binding cleft. Now with class 2, we see both alpha and beta chains, and like I mentioned, both of those are encoded for by the MHC class 2 gene. So they have an alpha chain and a beta chain, which then come together to form a heterodimer, and the peptide binding cleft for MHC class 2 is right in the middle, right in between the alpha 1 and the beta 1 loops. So here's a side-by-side comparison of MHC class 1 and MHC class 2. Remember that MHC class 1 will bind to smaller amino acid chains, 8 to 11 amino acids long, versus MHC class 2, which tends to bind larger chains of 10 to 30 amino acids in length. Remember that MHC class 1 uses cytosolic antigens or presents endogenous peptides, where MHC class 2 presents extracellular or exogenous peptides. MHC class I is expressed by all nucleated cells. MHC class II is expressed by dendritic cells, macrophages, and B cells, the APCs. And MHC class I, remember, is associated and recognized by CD8s, whereas MHC class II is recognized by CD4 T cells. So another question we have here, a 54-year-old man on dialysis for 1 year, secondary to polycystic kidney disease, undergoes a workup for a kidney transplant. In order to test for transplant compatibility, which proteins must be characterized? This is a K-type question again, meaning that there are multiple answers. Is it A, MHC class I, B, MHC class II, C, beta-2 microglobulin, D, beta chain, or E, chromosome 6? The correct answers are A and B, MHC class I and MHC class II. In fact, to be a little more specific, in class 1, the two that they really look at are A and B. And in class 2, the one that they really look at is DR. So A, B, and DR are the three regions that they focus on for transplant compatibility. So if you hear the transplant physicians talking about 4 out of 6 match, 5 out of 6 match, 6 out of 6 match, they're talking about the 2 A's, the 2 B's, and the 2 DR alleles. Beta-2 microglobulin, as we mentioned, is something that's constant. It's the same for everybody. Beta chain would not be specific enough in order to test for compatibility, and chromosome 6 would be far too broad. Here's an example of autosomal dominant polycystic kidney disease. You can see a normal kidney in the middle, and you can see on the outsides 2 examples of polycystic kidney disease. Note the huge cysts that take up almost the entire parenchyma and really expand these kidneys out. This concludes our discussion of MHC. In the next section, we will discuss humoral immunity.